Test 68C, Science Practice Problem number 1. Number 1 says, according to Figure 1, as the concentration of drug A increased, the percent of colony formation for cell Z did what? First thing we want to do is go to Figure 1, and we want to look at the concentration of drug A. So when we come down over here, we're going to compare that to the cell line Z. Here is our Figure 1 down here, and we see that in the x-axis we have drug A concentration, and we are starting here at 0, and we're moving over to the right and increasing the concentration from 0 to 100. So it's increasing. Now what we have to do is find cell Z. Cell Z is here. It's this region of the bar graph. When you come over here, you see here is cell Z right here. Here is Z. Here is Z. And so forth, all the way down. You can see as we move over from 0 to 100, the concentration is increasing, and the formation of the colonies is decreasing. It's a negative relationship. The correct answer, therefore, is going to be decreasing, which is answer choice B for number 1. Test 68C, Science Practice Problem number 2. Number 2 states, based on Table 1, cell W was found from tissue found in which system of the human body? First thing you want to do here is go up to Table 1 and find cell W. It's right here. And then it says organ of origin is the colon, and then you want to Scooby-Doo that into the answer. The colon is from the digestive system. Number 2, G. Test 68C, Science Practice Problem number 3. Number 3 states, based on Figure 1, if cells from cell line X had been treated with drug A at a concentration of 200 milligrams per liter, the percent colony formation would most likely have been what? First things first, we need to go down to Figure 1. Figure 1 is right here. Second thing we need to do is, if you look in the x-axis, you see the concentration is down here. The y-axis here is the percent colony formation. Here's the key. We are looking for cell line X. Here is cell line X here. It is the shaded region with the lines through it. If you look over here in 0%, here is cell line X here. Here it is here. Here it is here. And so forth as we continue on down. Now, if you look here, the number one thing ACT likes to ask you is relationship questions. If you look here, you see that this trend is going down. And this is going from 0 to 100. So as we increase the concentration of drug A, the cell formation is going down. It's a negative relationship. It can be represented with drug increase, formation down, plus minus. Here we see that in 100 milliliters, 100 grams per milliliter, that the formation of cell line X is 40%. So we know that based upon the trend, that the answer has to be 40% or less based upon the trend we've seen. If you look at our answer choice, the correct answer choice falls right in your lap, answer choice A, less than 40% based upon what you've seen. Remember, all science is about is looking for relationships. What happens when we do this? If we do this and we see that continually, if we do that, will the trend continue? Answer choice B is incorrect because it says 40 to 50. If we were to go 40 to 50, that trend would come up. Just like answer choice C, 50 to 60 and greater than 60. That's not the trend we see. We see the trend going down. There would be a spike here if any one of these answer choices were to be correct, B, C, and D. That's not the case. So 3, the correct answer is going to be answer choice A. Remember, kids, relationships, the most important thing, not only on ACT science, but in science in general. Test 68C, Science Practice Problem number 4. Number 4 states, according to Table 1, the concentration of drug A necessary to kill 50% of the cells from cell line Y was approximately 10 times the concentration of which one of these other cell lines? Now, first things first, kids, this is a long question stem that's 4 lines long. Make sure you put this in your own words and reword it. Make it easier. We're going to need to go to Table 1 and we're going to go ahead and find cell line Y. Then, which one of those answer choices below is 10 times less than cell line Y? See what I did? I just put it in my own words. Let's scroll on back up to Table 1. In Table 1, we are going to go ahead and find cell line Y. Cell line Y is here, and it looks like in order to kill 50% LD50, it's 70 milligrams per liter. 70 is 10 times more than which one of these numbers? Well, 6.9 is close to 7. 7 times 10 is 70, so that means the correct answer is going to be W. How did we know that that was going to be the 50%? If you look up here and you're not sure, in the table, LD50 is defined up here. LD50, the definition, the concentration necessary to kill 50% of the cells. If you see something here and you feel a little shaky with it, go back up into the mini paragraph and find the definition if you need to. Otherwise, cell W is the answer we should be looking for this particular problem. When you come over here for 4, you see that G is the correct answer. Test 68C, Science Practice Problem number 5. Number 5 states, at which of the following concentrations was drug A completely ineffective in preventing the surviving cell line V cells from dividing to form colonies? The key word here is completely ineffective. What we need to do is we need to go over here to figure 1. Then the second thing we need to do is we need to come over and find out cell line V. In the key, we'll always be using the key. Cell line V is here. It's the lightly shaded region. So we're looking for something that's completely ineffective. That means when they gave a certain amount of the drug concentration A, nothing happened. So we're going to go ahead and look over here. Percent colony formation. When it's totally ineffective, 100% of the colony formation will happen. If you look over here, here is cell line V again. Lightly shaded region is right here. And you see that at 0%, it is 100% colony formation. We look over here, 0.1. Here is the line we're looking for, V. And you see again, we're still at 100%. You look over here at 1 in the x-axis for drug concentration A. Then we find cell line V, which is here. It's still at 100%. Then we come over into the next one at 10 milligrams per liter. We see finally it starts working here around 78%. So we know the correct answer is going to entail the first 3 bars right here. So we look over here and see what those 3 bars are. Those 3 bars are 0 to 1. The correct answer, therefore, is going to be A, 1 milligram per liter. Test 68C, Science, practice problem number 6. Number 6 states, the ratio of tall offspring to short offspring in cross 1 is closest to which of the following? First thing we need to do is we need to go ahead and look for tall versus short ratio in cross 1. So let's go ahead and find cross 1. Cross 1 is here. And in cross 1, you'll see that we need to look at table 1. The first thing we need to do is we need to look at the ratio for tall. Here is tall right here. That will give you 154. So I just simply wrote it here and then put a dot, dot. And now we are looking for the short, which is here, and that will give you 46. So I wrote 46 here. 154 to 46, when you reduce it down, the correct answer is going to be H, 3 to 1. Test 68C, Science, practice problem number 7. Number 7 states, what is the genotype for gene T in the offspring from cross 2? First thing we need to do is scroll on down and take a look at cross 2, and we're looking for gene T's genotype. So we're going to go ahead and find cross 2 is right here. And when we look at cross 2, let's quickly glance at table 2, and we don't see anything about gene T in here. It just says number of offspring and flower phenotype. There's nothing about T in here. So what we have to do is go ahead now and read this little mini intro paragraph. 2 pink flowers, species X plants, each with the genotype RR. All right, close. We're crossed. And the numbers and phenotypes of the resulting offspring are shown in Table 2. That's all the information they give us. It doesn't say anything in cross 2 about gene T. So we know that the correct answer is going to be D. It cannot be determined. Simply just go to cross 2, look for information about the genotype T, and you see that there's nothing in there. 7 is D. Test 68C, Science, Practice Problem number 8. Number 8 reads, the percent of offspring from cross 2 with pink flowers was closest to which of the following? Go over here. First thing we do is find cross 2. We know that cross 2, there is Table 2 with information here. If you look over here, it says number of offspring. In the x-axis over here, column 1, and column 2, flower phenotype. Here are the colors, red, pink, and white, and here are the number of offspring. We're asked about the percent of pink flowers. We see over here pink, there was 102 that were pink, and now we need to figure out how many were there total. That means 46 plus 102 plus 52 is 200 total, and 102 of the 200 were pink. 102 divided by 200 is roughly 50%. That will give you the correct answer of H for problem 8. Test 68C, Science, Practice Problem number 9. Number 9 reads, suppose a gardener wants to produce tall pink flowered species X plants based on the results from cross 3. Which of the following pairs of plants, if crossed, would produce offspring with this phenotype? First things first, kids. Remember, whenever you get a question stem that's longer than 2 lines long, you need to put this in your own words. Get your pen or pencil moving and start circling stuff that you know you're going to need. First of all, we know we need to look at cross 3, so we have to find the right experiment. Number 2, we are looking for tall pink flowers. Then we need to find out what the genotype is, one of these answer choices, that will produce that. See what I did? It's basically putting it in your own words. Now, let's go ahead, get started. We're going to jump on down and take a look at cross 3. Here in the experiment, we find cross 3, and we notice that in cross 3, table 3 is the information we're looking for. We're looking for tall pink flowers. What is the genotype makeup for that? When we come over here, we see that here is TTRR. They're all capitalized. And when we have capital T and capital R, which are dominant genes or alleles, we see that we have a tall phenotype and we have a red phenotype. That tells us that TT is a dominant gene And the dominant production phenotype is going to be tall. So we can assume that small t's will produce short. Let's spot check our work. Small t's here, do they produce short? Yes, they do. Here we see TT again, small, small, in both 19 and 10 over here. And once again, you see that that's confirmed by short stems. When you come over here and you see RR is going to produce the dominant trait of red flowers. When you come over here, here is another RR. Make sure that we're on the right page. Is that producing red? Yes, it is. Over here, we see RR. And once again, we see red. Now, if we want to look for small RR, we see that that produces white. So we're looking for pink. Here's what we need to do. What produces pink? You come over here and see that the phenotype here, pink, is produced by capital R, small r. So we know if we want to get the color pink, we have to have a capital R and a small r. If we want to get tall, we need to have two capital T's. So the correct answer here is going to give us capital T, R, capital, small r. That will produce pink. If you look up at the answer choice and see what we did here, when you cross these two here, capital T is dominant trait, which will produce tall. Now, we need to combine to get capital R and small r. So if you take this one and combine it with the small one, you get capital R, small r, and you combine this one, capital R and small r, with this one, you get R, R. And remember from the table below, capital T, R, capital, small r right here is the genotype that will produce pink. The correct answer, therefore, is going to be answer choice A. Remember, kids, you may need this information. This is basic human genetics or animal biology genetics as well. It's all the same thing. We're going to need to know that capital letters are going to be dominant genes and small letters are going to be recessive genes. Dominant will always win over in the recessive gene pool. 9 is answer choice A. Test 68C, Science Practice Problem number 10. Number 10 states, a student had hypothesized that the majority of the offspring from cross 3 would be tall and have white flowers. Are the results shown in Table 3 consistent with this hypothesis? Once again, kids, it's a question stem that's longer than 2 lines long. Put it in your own words. We basically need to go find cross 3 and look in cross 3. Here is cross 3, and you'll notice that cross 3 contains table 3. We are looking to see tall and white. So come over here and find tall white. Here's tall red, tall pink, tall white. And we see that we have 11 offspring. Keep going. Tall red, tall pink, tall white again. And there's 20 more. Keep going, short, short, short. 11 and 20 make 31. and look at the rest of the numbers in here, 10, 18, 19, 41, 12, 19, and 10, is this consistent with what you see in the table? The answer is no, because 41 is the dominant one, which is tall pink. 41 of the offspring were tall pink. Here, this student said the majority of them would be white with tall. That's not true, because we saw 41 over here, tall pink. So the answer is going to be no. We can go ahead and cross out answer choice F because it says yes, answer choice G because it says yes. Answer choice H and J are closer. Let's go ahead and check them. H says no, only 11 offspring from cross 3 were tall with white. You know that that's wrong, because here is 11, but also we see here is 20. Tall white, tall white. 11 and 20 makes 31, so that is wrong. Answer choice J says no, only 31 offspring from cross 3 were tall with white. That's the correct answer. Remember, don't stop once you find one answer here. You got to look at the whole graph. You got to look at the whole chart. Make sure that you don't stop here. Some kids might have gotten this problem wrong and chose answer choice H because they didn't continue looking at the full chart. The correct answer for 10 is J. Test 68C, Science, Practice Problem number 11. Number 11, bear with me kids, this is a tough problem. We are going to make this extremely easy. Here's what 11 says. Suppose a short red-flowered species X plant is crossed with a short pink-flowered species X plant. The percent of the resulting offspring with the genotype small t, large R, small r will most likely be closest to which of the following? First things first, when you come over here, a quick review of some genetics. Whenever you have a capital T and a capital T, these are considered dominant genes. How do you know that? Here is capital T genotype here, and you see that that is going to produce a tall plant. We are looking for short. They give us this information already. Two short plants has to be two small t's. So now this information becomes non-significant here. This is where we need to start focusing. Red and pink flowers. Well, if you look over here in the genotype, in order to get a red flower, you have to have two capital R's. That is a dominant gene. On the other hand, if you need to get a white flower, which is a recessive gene, you need two small r's. We are given pink. So that means we have capital R, small r. It's a mixture of dominant and recessive genes. Now, some of you are already starting to scratch your head. What is he talking about? Watch how easy this is. First of all, we already know that the stem length here is short, and that has to be small t. So let's get rid of that. We're going to focus on capital R, small r. Now, if you come over here, I'm going to clear the screen and watch how easy this is. We're going to go into table 3 and we're going to find capital R, small r, and when we see that in the genotype here, let's look and see. There are 18 offspring with this genotype. We keep going down here, we find another one, capital R, small r. We see that that produces another 41 offspring. We keep going down here and we find another capital R, small r, and we see that that produces another 19. 18, 41, and 19 gives you 78 total with this genotype. 78 of what? Add this up. 10, 18, 11, 19, 41, 20, 12, 19, and 10 gives you 160. 78 is close to 80. 80 over 160 is half, or 50%. Based upon what we see, capital R and small r, that is going to lead us to the correct answer of C. Forget all this other stuff here. It's way too hard to explain. Just simply use the information from table 3 and calculate out 50% for 11. Test 68C, Science, Practice Problem number 11. Number 11, bear with me kids, this is a tough problem. We are going to make this extremely easy. Here's what 11 says. Suppose a short red flowered species X plant is crossed with a short pink flowered species X plant. The percent of the resulting offspring with the genotype small t, large r, small r will most likely be closest to which of the following? First things first, when you come over here, a quick review of some genetics. Whenever you have a capital T and a capital T, these are considered dominant genes. How do you know that? Here is capital T genotype here, and you see that that is going to produce a tall plant. We are looking for short. They give us this information already. Two short plants has to be two small t's. So now this information becomes non-significant here. This is where we need to start focusing. Red and pink flowers. Well, if you look over here in the genotype, in order to get a red flower, you have to have two capital R's. That is a dominant gene. On the other hand, if you need to get a white flower, which is a recessive gene, you need two small r's. We are given pink. So that means we have capital R, small r. It's a mixture of dominant and recessive genes. Now, some of you are already starting to scratch your head. What is he talking about? Watch how easy this is. First of all, we already know that the stem length here is short, and that has to be small t. So let's get rid of that. We're going to focus on capital R, small r. Now, if you come over here, I'm going to clear the screen and watch how easy this is. We're going to go into table 3 and we're going to find capital R, small r, and when we see that in the genotype here, let's look and see. There are 18 offspring with this genotype. We keep going down here, we find another one, capital R, small r. We see that that produces another 41 offspring. We keep going down here and we find another capital R, small r, and we see that that produces another 19. 18, 41, and 19 gives you 78 total with this genotype. 78 of what? Add this up. 10, 18, 11, 19, 41, 20, 12, 19, and 10 gives you 160. 78 is close to 80. 80 over 160 is half, or 50%. based upon what we see, capital R and small r, that is going to lead us to the correct answer of C. Forget all this other stuff here. It's way too hard to explain. Just simply use the information from table 3 and calculate out 50% for 11. Test 68C, Science, Practice Problem number 12. Number 12 states, which of the following diagrams of the side view of the beam correctly portrays the directions of the three forces that the apparatus exerted on the beam? First thing we want to do is we want to go up and look at the diagram of the apparatus. So let's scroll on back up and take a look at Figure 2. Figure 2 is a diagram of the whole experiment here, and you'll notice that here we have the beam, and then you have a platform here with the weight, which they call the load. Then that is balanced on this little rod, and then the force is measured here from this. So when you put weight on a beam here, you'll notice that the beam is going to be deformed like this. If you look, you can see that down below in Figure 3. It even shows you that. Here's the side of the beam. The weight would be right here in the middle, and the board or beam is going to be deformed once you put the workload on there. So where are the forces? The forces are going down here. As you put pressure here in the middle, the force is going down, but also this is starting to spring up. How do you know that? Because you need some type of force to go from straight here to raise it this amount. So you know there is going to be force going up as well. If you didn't have that force here, it would still be straight. So, quite simple, you have force going up on the edges and force going down in the middle. When you look at the diagrams here to see which one of those answer choices gives you the best description of that, you see that G is the only answer that's going to work. Here is the workload in the middle pushing down, and then the beam is going to curve up at the edges here. So there'll be force there and here. 12 is G. Test 68C, Science, Practice Problem number 13. Number 13 reads, if in study 3 a trial had been conducted in which W equals 25 newtons, the D would most likely have been closest to which of the following? First thing we need to do is go into study 3 and take the new value for W at 25. Let's go back up and take a look at study 3. When you look at study 3, we are going to be in table 3. Here's study 3, table 3. We have a new W value of 25. So you simply look, find where 25 under the W column would be. Here's 20, here's 30. It would be right between here. It would be between 10 and 11 trials. Go ahead and look what the D value would be. The D value would be between 4.2 and the 30W, which is trial 11, 6.3. What's between 4 and 6? How about 5? 4, 5, 6. 5.1. Seems like a reasonable number. We previewed our answer. We go back to the question and look for an answer that says right around 5 times 10 to the negative 6. We scroll on down, and there it is right there. 5.2 times 10 to the negative 6 meters. 13, guesstimation. Test 68C, Science, practice problem number 14. Number 14 states, if the amount of work done to deform a beam equals W times D, in which of the following trials was the amount of work the greatest? First thing we need to do here is we need to go back into the mini introductory paragraph and start defining some terms. If you notice that when we were in here, it told us we need to go to several different experiments, 2, 4, 6, and 8. So right away, if you look and find where experiments 2 and 4 are, you'll see that they are in study 1, and we can locate that information in table 1. Here's table 1, and here is trial 2 and trial 4, and we're dealing with I, and we are dealing with D. So let's go ahead and remember what I and D are. So if you come into the mini introductory paragraph, it says here, in the mini introductory paragraph, it says the beam was subjected to a load, W. So that means that this W is going to be the weight. Then it will give us a definition for D. It says a dial on the apparatus registered the distance. And then down here at the bottom, it gives us our I value, and the I value is the effect of the width and height of the beam. I is basically just the size of the beam, basically. So when you come over here, we've got in study 1, over here you have basically how big the beam was and the distance that the board was transformed. So the bigger the beam, what happens to the distance it was transformed? 2 to 4, 8 to 1. You see that these arrows are going opposite, which makes total sense. The bigger the beam, the less it's going to be deformed by the weight. So, if work is defined as they say down here in the question, the weight times the distance, you know right away when you look at trial 2 and 4, you see that the distance that it's transformed is going to have a higher value. You know that you're going to have here a big board and it's going to be less transformed, so there will be less work done. Trial 2 is going to be a greater value for W times D. Now we need to go down and look at the next two experiments and compare those. Remember, now we're going to look at trial 6 and trial 8. Let's go ahead and find those. And trial 6 is here. And trial 8 is here. And now we're dealing with E. E and distance. So if you look over here, if you remember what E is, E from the mini introductory paragraph is here. And E says it's the stiffness. Well, a stiff board is going to bend less when you put weight on it. That's just common sense. So a higher stiff board, the larger the number here, the more stiff the board is going to be, or beam, the less it's going to travel. And that's what you see here. This is a very stiff board, and you see over here that the distance it was deformed was not very much. Here is the least stiffest board, and you see that the distance was larger, which means the beam was bent more. If we were to draw some diagrams here, you would see that this board probably would look something like this, and this board or beam would look probably something like this. Which one is deformed the most? Here, we're asked about trial 6, trial 8. You see there's a direct correlation between the distance it's deformed and the amount of work that we have. So right away, we are going to cross out answer choice that contains 8. And now we need to compare trial 6 from study 2 with the trial from study 1, which was study 2. Compare the distance here. Trial 2 is 2.1 times 10 to the negative 6. It was deformed. Or study 6, the distance it was transformed or deformed, you see, was greater, 4.2. Remember, this distance, they said, is going to be the amount of the board transforming down into a curve. Which one is greater? 4.2. As we see in 6 or 2.1, you can see that this number is greater. So therefore, the best answer choice here when you put it all together is going to be trial 6. It's combining results from several different places. Remember, if it gives you terms like E, D, and I, you have to go back in there, take a look, and make sure you know what you're dealing with. This particular example, the correct answer is going to be trial 6, which leads you to answer choice H. Test 68C, Science, Practice Problem number 15. Number 15 says, the results of study 1 are best represented by which of the following graphs? First thing we need to do here is we need to go ahead and take a look at study 1 and see what we're dealing with. If you look at answer choice A, B, C, and D, you notice that the y-axis on every single one of them deals with D times 10 to the negative 6th meters. Now, if you look at answer choice A, in the x-axis, we are dealing with E. In B, we are dealing with E. In C, we are now dealing with I. And D, we are also dealing with I. So the first thing we want to do is go up to study 1 and go to the right place, are we dealing with I or are we dealing with E? Let's go on up, take a look at study 1. Study 1, we can go ahead and look at table 1, which is under study 1 here. Study 1, table 1. And in table 1, we are dealing with I. So that means right away, we can come back over here and we can cross out answer choices that entail E. That means answer choice A right away can be crossed out because the x-axis is wrong. We're not dealing with E in study 1. That is called a wires crossed problem. A is out. B can be crossed out for the same reason. E, the x-axis is E. We are looking for I. Now, that leaves us with answer choice C and D. If you look at answer choice C, we look like we are increasing the x-axis from 0 to 10. And as we increase going from 0 to 10, it looks like our y-axis is going from 0 to 5. This is a line, like a staircase, going straight up. And you remember from the mini lecture that we had in the one-on-one tutoring, this is a positive or direct relationship. If you look at answer choice D, we go from 0 to 10, which is increasing. But now we're going from 0 to 5. The numbers are high and going from high to low. Reminds you of a down staircase. And a down staircase from the mini lecture is always going to be a negative or indirect relationship. We have to go ahead and look in table 1 and see what the relationship is. Remember, when you have a chart of numbers with columns in there, you can look at the chart of numbers and put arrows going in the direction to see what kind of relationship you have. Let's go on back up to study 1, take a look at table 1 and see what we see. When we scroll back up to table 1, you will look and see that when we're dealing with I versus D, as I increases, what happens to D? D, the numbers go down. Small to large, large to small. The arrows are going opposite, so you know this is a negative relationship. As we come back over here, the correct answer here is going to have to be D. As you increase the I, the D is going to be going down. It's a relationship question. D for practice problem 14. Test 68C, Science, practice problem number 16. Number 16 states, the beam tested in study 3 was most likely composed of which of the following metals tested in study 2? First thing we want to do here is we're going to have to go ahead and take a look at study 2 and study 3. Go back up here and we're trying to figure out which metal was used. So if you go to study 2, let's go ahead and take a look and see what is going on here in study 2. Scroll on back up to study 2. Here is study 2, and it tells us that there are various metals, S through V. Okay? Now it says in every trial the workload equaled 20 newtons. Let's go on down to study 3 and look for a workload of 20 newtons. You scroll into study 3. We come over here and we find work equals 20 newtons. under the work column. And you see that the distance the work was transformed equals 4.2. Now we can take this information and go on back up to study 2 and find 4.2 for the distance. Study 2, here's study 2 again. We know that the distance it was transformed was 4.2. So we come over here into the column that says distance, find 4.2, which is right here. And we see that that metal is metal T. Take the information from study 2, throw it into study 3 to get another number, which is the D value. Throw the D value back into study 2 to find out what metal it was, and that will lead you to T. The correct answer on that is going to be answer choice G for 16. Test 68C, Science, Practice Problem number 17. Number 17 states, based on the results of study 1 and 2, for a given W, which of the following combinations of I and E would yield the stiffest beam? First thing we need to do is, kids, go back in, put in your own words a definition for I and for E. Then we need to go back into study 1 and 2 and look at those tables and look at the arrows that we wrote in the columns. Remember, when you have a chart full of numbers, put arrows next to the numbers to represent the relationship. As the set of numbers here goes down, does this go up or down? Use your arrows to help out. Let's go ahead and basically finger scan for the definition of I and E. When we do that, we'll find that in the mini introductory paragraph. Starting at the top, if you were to go ahead and finger scan, let's start for I, you would basically come over here, I. Keep going until you found I defined. You'll notice that that information can be found down here. And what it says about I is this. The effect of the width and the height of the beam, C in figure 1, on D, represented by the quality of I. The height and width of the beam. So really what this means is I is the size of the beam. See what I did? I put it in my own words, and now it makes more sense. Then we finger scan for the definition of E. We find E here, and it tells us it's Young's modulus, which is the stiffness. I, size of the beam, and E is the stiffness. When we go over here, let's go into study 1. And you see in study 1, we're dealing with I, which is the size, shape of the beam. And you'll notice that we put our arrows in as we increase the size and shape of the beam. The distance it's transformed goes down. That means a small, less thick beam is going to bend more. As you increase a thicker, larger beam, it's going to bend less. Let's go into study 2. In study 2 here, we are dealing with E. E is the stiffness. As you increase the stiffness, the distance it's transforming or deforming goes down. That makes sense. A thicker board that's stiffer is not going to bend as much. See what I did? I combined the results here. Stiffness and the size of the board. You know that both of these are increasing the stiffness, and the size will transform less. So when you come over here, in the answers, which one, what are we looking for here? which one would yield the stiffest beam? The stiffest beam would have the highest E, which is the size, 3344, and the E is the stiffness. The stiffness would be the greatest, so you have 40 here or 40 here. The greater I and the greater E combined, here is the greatest stiffness for I, here is the greatest stiffness for E. The correct answer when you combined stiffness and the size of the board, both are going to be the largest, which will produce answer choice D, 4.0 and 40 for 17. Test 68C, Science, Practice Problem number 18. Number 18 reads, in experiment 3, which of the biodiesels has a TBHQ concentration of 750 milligrams per kilogram decomposed most quickly in the AOA? First thing you want to do here is don't get confused. It's merely just going and taking a look at these experiments and putting the numbers in place. Let's go ahead. We're going to take a look at experiment 3, 750 milligrams per kilogram. We scroll back on up to take a look at experiment 3, and you see that here is experiment 3, and experiment 3 deals with figure 3. And in figure 3 now, we want to go ahead and take a look at the x-axis. Here is the concentration of TBHQ, and here is the induction period. The induction period means it's the amount of time that it will take to decompose. So as you increase from 0 to 100, it's taking longer for this biodiesel fuel to decompose. Now, we are asked in this question about 750. Here is 750 here. We are looking for the answer to come out of this column here. Let's go back and take a look at what they're asking us one more time. It says here, which of the biodiesel fuels has the concentration of 750 that will decompose the quickest? Which one of these will decompose the fastest? Now, it's a matter of just looking back at figure 3 in the 750 column and realizing that the induction period means decomposition, decomposing. So which one decomposes the fastest? It's the smallest one here, which is the black bar. We come over here and find in the key what the black bar means. The black bar is BD3. Go back down to the answer choices and see that the correct answer, BD3, is found only in answer choice H, 18H. Test 68C, Science, Practice Problem number 19. Number 19 says, a chemist claims that if an antioxidant concentration in a biodiesel fuel is increased from 500 milligrams per kilogram to 1,000 milligrams per kilogram, the biodiesel's fuel stability will increase. The claim is inconsistent with the results in experiment 3 for which biodiesel fuel. First things first, we need to jump on in and take a look at experiment 3. Now, experiment 3, this chemist says that as we increase from 500 to 1,000, the stability will increase. That means there's a direct relationship. Increase and increase, it's a direct relationship. We're looking for one of the answers or one of the biodiesel fuels that is inconsistent, that does not increase the stability as you increase the concentration. Let's jump on up, take a look at experiment 3. And when we find experiment 3 up here, here is experiment 3. You will notice that we are dealing with figure 3. And quick overview of figure 3. Here in the x-axis is the concentration. And here is the induction period. The induction period is how long it takes for this biodiesel fuel to decompose. Let's go ahead and take a look over here at the key. You always want to use a key. When they give you a key, always look at this. First one here is the shaded bar, which is BD1. You come over here, we're starting at 500, like you said, the chemist, and says as we increase from 500 to 1,000, we find that BD1, the shaded bar, is roughly about 74, and now at 1,000, when we increase the concentration, we see that this goes to 115. You simply draw an arrow here, and as you increase the concentration, it looks like the induction period increases. That's a positive relationship. We're looking for a negative relationship, one that is inconsistent. We can go ahead and cross out an answer choice that has BD1. If you look at the next one, it's got the striped bar. It's BD2. We go into 500, find the striped bar. We see it is roughly at 70 in the 500 concentration. When we move over to the 1,000 concentration, you see the striped bar, which is BD2, now increases to roughly 75. Draw an arrow here, and you see that as we increase from 500 to 1,000, the bar increases. That is a positive relationship, plus plus, which is what the chemist predicted. And we are looking for the one that's inconsistent that can be crossed out. We go to the next one and continue on with the same process. It's the black bar. It is BD3. 500 concentration, you find the black bar. You see that we are at about 75. We increase the concentration to 1,000 and see that now we're down here at 10. As we increase the concentration, you see the induction period goes down. As we increase the concentration, the induction period goes down. That is inconsistent with what the chemist predicted. Let's move on and take a look at the last one here, just to make sure we have the right answer. But so far, I like BD3. BD4 is the non-colored bar. If you look at 500 concentration, you see that we are just under 40 over here, about 39. Then we increase from 500 to 1,000. Find the non-colored bar over here. And now you see we are at 40. So it goes from 39 at 500 to roughly 40. That is still an increase, ever so slight, but that arrow is going in the same direction as BD1, BD2. That is a positive relationship. As we increase the concentration, the induction period goes up. The only one that's going to be inconsistent here is going to be the one with BD3. Let's go ahead, scroll down to our answer choices here, and take a look at one that gives us that answer. BD3 is found in answer choice C for number 19. Test 68C, Science, practice problem number 20. Number 20 states, suppose a set of trials have been done in experiment 1 at 115 degrees Celsius. Which of the following would have been the most likely induction periods for BD2 and BD3? So the first thing we want to do here is go to experiment 1, find 115 degrees. Then we're looking for BD2 and BD3. So let's go back up to experiment number 1, and you'll see that experiment number 1 is here, and experiment number 1 is dealing with figure 1 here. So we are going to find the new temperature of 115 degrees here in the x-axis is temperature. Here's 100, 110, 120. So 115 degrees will be right here. The first thing is we're looking for BD2. So BD2 at 110 is the stripes. So that is right here. And you see that we are 20, 40, this is 30, that's probably 28 or 29. Then if you look over here, the stripes here would be 20. Since we're halfway in between 10 and 20 at 115, we know that would probably be right about in the middle. Distance between 28 and 20 is 4, so 20 plus 4 would give us this BD2 around 24. And now we'll do the same thing for BD3. BD3 is here at 110. It's the black bar. You see that right here we're at 40. And when we move up to 120, you see we are down here now at 30. Halfway in between 30 and 40. And the new value would be 35. So the answer would say something like BD2 right around 24, 25, and BD3 35. Let's go back down to the answer choices and see that the correct answer here is BD2 is 25 minutes and BD3 is 35 minutes. Correct answer for 20, F. Test 68C, Science, practice problem number 21. Number 21 states, a sample of fresh BD1 is tested in AOA in experiment 1 and found to have an induction period of 65 minutes. At which of the following temperatures was the test most likely conducted? First things first, kids, make sure you circle the information we're going to need. We need to go to the correct experiment, that's experiment 1. Then we need to find in the key which bar is BD1. Then we need to predict 65 minutes what that temperature is going to get. Let's go back up to experiment 1, and when you see experiment 1, you see that experiment 1 is located in figure 1 here. Let's do a quick overview of figure 1. Here is temperature on the x-axis and induction period in minutes for your time on the y. Here is the key up here. And we're looking for, once again, BD1, which is the gray bar. Now, let's do a quick overview of what we see here to kind of get about understanding the relationships. As we go from 100 to 130, the temperature is increasing. So let's put an arrow going up. As the temperature increases from 100 to 130, notice that these bars go down. So it's a negative relationship. Here's what we see. if you look at the one we're looking for is BD1, the shaded region here. You see we're right here, and if you go right across here, you see we're roughly at 30. Now let's go down to 100 and look for the same thing. You see here we're roughly at 60. The difference between 60 and 30 is 30 degrees. 30 degrees, what happened between 100 and 130? Well, we went from 60 minutes down to 30 minutes. 100 to 130. So 30 degrees equals 30 minutes. Let's reduce that down. That means 1 minute equals 1 degree. The information in the passage they give us is 60 minutes, but we want 65. So we need to increase 5 more minutes. Remember, this is an inverse relationship. As we increase the temp, the time goes down, or as we increase the time, the temp goes down. See that? This is where the arrows will come in handy. We're at 60, we want to go from 60 to 65, so we know that that means the temperature is going to go down at 1 degree per minute. We go from 60 to 65, we have 100, and now we need to go subtract 5 degrees off of that, which would give you 95. Remember, inverse relationship. Draw your arrows in there. As you go from 100 to 130, these time periods are going down just like you see here. We're going from 100 degrees, subtracting 5 because the time is going from 60 to 65. The correct answer, therefore, should be 95. Let's scroll down and take a look and see if an answer choice is close to that. And when we go back to 21, you see the correct answer here is going to be answer choice A. Test 68C, Science, Practice Problem number 22. Number 22 reads, in experiment 1 through 3, the component of the air bubbling through the biodiesel sample that was primarily responsible for the breakdown of the sample was what? Air bubbling. Scooby-Doo this into one of the answer choices. What is air bubbling? Is air bubbling H2O or water? No. Is air bubbling hydrogen, H2? No. Is air made up of N2? No. Is air made up of O2? Yes. Air bubbling is Scooby-Dooed for O2 oxygen. 22, J is the correct answer. Another way that you can get this is to go into the mini paragraph all the way back up at the top, and it says right here in the first line, it says this: when an oil is exposed to air, small amounts of the reactant peroxides can form in the oil. So they are going to basically shoot air in there to produce a peroxide. Exposed to air, O2. 22, J. Test 68C, Science, Practice Problem number 23. Number 23 reads, suppose BD5 is made by mixing 250 milliliters of BD1 with 750 milliliters of BD4. If a sample of BD5 contains 500 milligrams per kilogram of TBCA that was tested in experiment 2, its induction period would most likely be. First things first, kids, this is a long question stem. You need to go here and put this in your own words. We need to go back up in and take a look at where they're dealing with TBCA. Then we're looking, first of all, let's go ahead and do that. So if you see, we go back up to the experiments, you see that we're going to find TBCA here in figure 2, experiment 2. Here is TBCA. So let's look and see what they're dealing with in experiment 2. It looks like the full concentration is 500 milligrams per kilogram. So to get a full load is 500 milligrams. All right, so we're in experiment 2. Now, the second thing that we're going to do here is we're going to go ahead and take a look. We are going to make a new solution, which is called BD5, from BD1, which is 250, right? So let's go back up and find BD1 and see what that is. So we'll go back to figure 2, and under TBCA we find BD1, which is here. Then we go ahead and it's the lightly shaded. We see that that's right here, and we come across and see that we are at an induction period of 85 minutes. So let's go ahead and write that down. So we have BD1 is 85 minutes times, how much are they going to use? 250 milliliters. So let's go ahead and say 0.25. 0.25 times 85 gives you roughly 21 minutes. Then let's go ahead and do the same thing over here with BD4. Now BD4 is 750 milliliters. So let's go back up into experiment 2, figure 2, and under the column antioxidant TBCA, we're going to go ahead and find BD4, which is the non-shaded bar. It's right here. And now we're going to come across here and see we are at 20 minutes. $20 minutes times 750 milliliters, or times 0.75, because 0.25 and 0.75 will give us a whole round number of the full amount. So 20 minutes times 0.75 will give us 15 minutes. So that means if we just had BD4, it would give you 15 minutes, but we're making a new solution, which is BD5. It's a combination between BD4 and BD1. So BD4 is 20 minutes at 0.75, 15 minutes. Then we're going to add that to the 85 minutes of BD1 at 0.25, which gave us 21 minutes. We add both of those together here. 21 minutes from BD1 here, and 15 minutes from BD4. We add those together. That gives us roughly 36 minutes. The answer is going to have to say something between right around 36 minutes. You look at your answer choices. A can be crossed out. It says less than 20 minutes. B says between 20 and 40. We derived 36. That works because it's right in there. C says between 40 and 80. That's out. And D says greater than 80. That is out as well. The correct answer, therefore, is going to be answer choice B. Now remember, don't get confused with this, because up here in experiment 2 says that we're dealing with 500 milligrams per kilogram. We just calculated one full load percentage-wise. Don't get confused with that. The percentage 0.75 and 0.25 will give you the full amount, which is one full load. That's how we got the 0.25 and 0.75. Think of it as like moles per liter or grams per mole. So when you multiply that percentage, because remember 0.75 is 750. We're going off of 1,000. So the correct answer here is going to be answer choice B. If you have any further questions about this particular problem, please see one of the Junior Genius tutors or talk to Professor Thomas to go over this problem. Once again, 23 is B. Test 68C, Science, practice problem number 24. Number 24 states, assume that the volume of water in each wetland remained constant over 120 days of the study. According to figure 2, 120 days after the allicor was added, what percent of the original allicor concentration remained in E2? First thing we want to do is this. Go find figure 2, look for E2 at 120 days. Then compare that amount to the original amount that was in there. Let's go ahead and jump on up and take a look at figure 2. Figure 2 is right here. And from the key, we'll always use the key. We're looking for E2, which is rectangle. And the rectangle here is right there. And you can see that that is roughly, 1%, 2%, somewhere around there. So here is the original concentration of 25 grams per liter. And now we have 1 gram per liter. So 1 over 25 gives you 1/25, which is less than 20%. It's the only answer falls in our lap when we look at our answer choices. Less than 20%, because remember the original amount was 25 grams per milliliter, 100%, and we are less than 5 grams, right around 1 or 2 grams. F for 24. Test 68C, Science, practice problem number 25. Number 25 states, according to figure 1, 50 days after ultrazine was added, its concentration in the water in S1 compared to its concentration in the water of E1 was what? First things first, circle the information that we need. We need to go to figure 1. That's right here. Second thing is we need to talk about E1 and S1. Circle them. Here's the key. I took this information and went ahead and just made it easier to see and just simply labeled it. We need to know about E1. So E1 is the rectangle right here. So I just basically wrote it in, E1. And S1 is the triangles. Triangles are up here. So here is our S1. Then what we need to do is add in the last bit of information, 50. Come over here in the XY axis, find the X days, find 50, here's 40, 60, halfway in between is 50, and find your numbers. E1 produces 10. E1 is 10. And our S1 value, when we go straight up here, is about 16. The next thing we need to do is we need to make sure that we word this into something that we understand. Read it one more time. According to figure 1, 50 days after atrazine is added, the concentration. The water in S1 is what? The water, is it higher or is it lower? Come over here and see S1. We said here it was 16. S1 is higher than E1. So right away we can cross out answer A and B, and you see the difference between S1, 16, and E1, 10, is about 6. You look at your answer choices and see that C is the closest answer choice based upon what we kind of guesstimated here. Remember, kids, a quick overview here. Circle what's important from the question stem. The second thing is if you get something from the key in some type of a chart or graph like this, don't be shy. Go ahead and write on your test and write this in here so you don't get confused. They're trying to confuse you with different lines. Make it as easy as possible and use your pencil to write on your test. The correct answer for 25, C. Test 68C, Science, practice problem number 26. Number 26 reads, according to figure 1 and 3, in S1 as atrazine concentration decreased, what happened to the DEA concentration? This is a relationship question. Let's go back in and take a look at figure 1 and figure 3. When we scroll back up to figure 1, you see we're looking for S1, which is the triangle. And we see that as we increase the x-axis from 0 days all the way to 120 days, the atrazine concentration goes down. This is an inverse relationship and can be represented by increase in days, atrazine concentration down. Another way it could be represented is as you increase the days, concentration goes down. Plus, minus, minus, plus, or the arrows. It is a negative relationship for figure 1. Now, let's go ahead and take a look at figure 3. Figure 3, the x-axis is days again, going from 0 to 120. And now we're talking about the DEA concentration. As we go from 0 to 120, it looks like the days are increasing. Relationships again. We can put our arrow like this. And what happens to the concentration? We go up. That is a direct relationship. Or we could use the plus-minus system. Either way will work. As you increase the days, concentration goes up. The other way you could look at this is the staircase that we talked about in the lecture. This is a classic example of a staircase going up, which is a direct relationship. If you looked over here in figure 1, you would see a staircase going down. It's the same thing. Positive and negative relationships. Here is your staircase, and you see the stairs are going down. So, you know that as you increase the concentration of ultrazine, the concentration is going to go up for DEA. If you were to increase the concentration of DEA, the concentration of S1 would go down. It's a negative relationship, so you know when we come back over here, read the question again so we get unconfused. In experiments figure 1 and 3, in S1, the ultrazine concentration decreased, then you know that the DEA is going to have to go up because of this negative relationship. F is the only answer that's going to work there. Remember, these arrows really are helpful, or the plus-minus system. 26, F. Test 68C, Science, practice problem number 27. Number 27 states, is the statement, over 120 days of the study, ultrazine concentration was most reduced in the water, in the open water wetland, supported in the data in figure 1. First thing you need to do here, kids, is this. Number 1, make sure that you translate this into your own words. Whenever you have a question stem that's longer than 2 lines long, it gets confusing, so we're going to have to get our pen or pencil moving and circle stuff. First of all, we need to do this. We need to go to figure 1 and now translate this in your own words. In figure 1, over this 120-day period, ultrazine concentration was most reduced. It was the least in open waters. Is that true? Let's go back in and take a look at figure 1. When you go up to figure 1, here is figure 1. We're going to need to take a look at the key. The key will tell us the different wetlands are here, and we are looking for open water. So open water is O1, which is the circle. You see here is the circle, and here is 120 days. So was it the least in this experiment? Here's the ultrazine concentration. Basically just trace the circled line, and was it the least? The answer is no, it's not the least. So we can go ahead, go back to our answer choices and cross out some stuff already. When you go over here, you can go ahead and cross out all the yeses. That means answer choice A is out because of yes, and B is out because of no. Now, let's look at the last 2 answer choices a little carefully. It says no, 120 days after ultrazine was added, its concentration was least in water in O1. Now, D says the exact opposite. No, 120 days after ultrazine was added, its concentration was least in water, E1. Now, we know that the correct answer has to be either C or D. The answer is no. We've seen that already. So what we need to do is we need to see where was it least. See what I'm constantly doing here, kids? Putting this stuff in my own words. Where was it least? Was it least in O1 or E1? Go back up here. Take a look at figure 1 again. Remember, we're looking for concentration at 120 days. Here is O1 here. And here is E1. Where was it the least? Well, O1 was the most. And you see E is down here. E1 is the least. So the correct answer is going to be least in E1. Let's go back down to the answer choice and see that answer choice D is the correct answer here. Remember, sometimes it's going to take you a couple passes to start crossing stuff out and then go back up there and look at it again. Once again, if you see a question stem that's longer than 2 lines long, put it in your own words and get your pencil circling things that are important. Try as much as you can to put it in your own words so you understand what you're doing. When you deal with science terms that you're unfamiliar with, you've got to put it in common sense terms so you know what to look for. 27, D. Test 68C, Science, Practice Problem number 28. Number 28 states, as shown in Figure 2, every time the alkyl concentration in the water in O2, S2, and E2 were measured during the study, the concentration in S2 and E2 were found to be very similar to the concentrations in O2. The most likely explanation for this result is that in S2 and E2, first things first, kids. Go ahead and ask Professor Thomas, what did you just read? The answer, I have no idea. Something about E2 and S2 being similar somehow. And oh yeah, in Figure 2. You have to go ahead and take the time to put this in your own words. Long question stems like this that are longer than 2 lines long, you will definitely get this wrong, because if you don't put it in your own words, you'll get it wrong. There's too much information here. So let's weed through this and try and figure out what's going on here. And it might take several passes for us to get this problem correct here. First of all, we need to go to Figure 2. And in Figure 2, we have to look at O2, S2, and E2 and see what's happening. So let's go ahead and do that. We'll get our feet wet. We'll go all the way back up here, and we'll look at Figure 2. Figure 2 is right here. And here is the x-axis, the days after the herbicide was added, and here is the concentration. And you'll notice that O2, S2, and E2 from the key are right here, and basically everything is doing the same thing. It's all the same. It's starting high at a concentration of 25 and going down out here to 120. Everything at about 40 days starts going around less than 2 grams per liter. This is pretty much the same thing. So they're all doing the same thing. Okay, great. Now, let's go back and weed through the rest of that question. It says, the concentration S2 and E2 were found to be very similar to O2. Yep, we saw that, right? The most likely explanation for the result is that S2 and E2 do what? Why are they similar? Okay, S2, E2, and O2, I'm still scratching my head just like you. I know you're scratching your head. Wait, what? What are you talking about? Let's go back up into the information from Table 1. And in Table 1, you'll see here, it gives you a definition of what all this monkey business means. What is S2, E2? It's hard to keep track of all that. So here's the wetland. Here is the O's. And that means O stands for open water. E, submergent. Okay? And if you look over here, that means that the plants are growing in the lake, but they're underwater. And here, emergent, E, means there's plants in the lake, but the plants are popping their head out of the top of the water. So, if you come back over here, this all does the same thing. So you know that the plants don't really have anything to do with the concentration and the days of the herbicide. The gist of the passage now becomes plants. Right away, when you look at your answer choices, you can go ahead and cross out H, because H says soil. That's not what S2 and E2 are about. J says soil. Cross that out. F and G are on track because it says plant uptake. Both of them. So, let's go back and do one last pass. In figure 2, concentration is all the same after 120 days and at 40 days. Do plants affect the concentration and the growth in this stuff? No. Plants don't have anything to do with it. There's something else going on. So when you come over here and look at your answers, you see that answer choice F says this. Plant uptake played a more significant role in the removal of allicor than did the absorption of the soil. That's not true. It did less, because if we had something that was significant, this chart would look different. If the plants did something to the concentration of this drug or herbicide, then the open water one would look different because the plants would have absorbed more. But in this case, it didn't. So that tells you that the plants didn't really do anything. Take a look at G. G says plant uptake played a less significant role in removing an allicor than did the absorption into the soil particles followed by bacterial decomposition. Plants didn't really have anything to do with this. The reason why, over here, we know that the difference between the experiments is S2 plants, E2 plants, O1 and 2 no plants. Do plants play a role? Yes or no? From this chart here, you know that plants do not play a role in the absorption of the concentration of allicor. So the correct answer for this one is G for 28. If you have any further questions about this particular problem, please see one of the Junior Genius Tuners or Professor Thomas at the Genius Combine. Test 68C, Science, Practice Problem number 29. Number 29 states, based on student 1's description of the ball's motion compared to the ball's kinetic energy at point A or point B, the ball's KE, kinetic energy, at point L is what? First things first, we need to know what KE is. Make sure that you understand this. You can find this information from the mini intro paragraph up here. You find student 1 is here, and we should find out what KE is. We can go ahead and finger scan up here for KE, and KE, the definition of KE is here. Here it is. Kinetic energy. It's 1/2 mass times the velocity squared. You come over here, it's basically like how fast it's going. Alright. Now when you come over here, it says, read the question one more time, now that we know that this is the kinetic energy, or kind of the, just think of it as almost like speed. Even though it's different, just think of it as speed to make it easier. Based on student 1's description, the ball's motion compared to the ball's kinetic energy at point A or point B, the ball's KE at point L is what? We need to look at student 1 and compare point L to point A and B. Now, we'll use a little bit of kid logic. We'll go into student 1. which is figure 1 here. Here's student 1. And here is figure 1 right here. And now we'll find point A and B. Here's point A. And here's point B. And think of this as like a string with a ball on it or a pendulum. If you're holding the ball up here, the ball is stopped. When you let go of the ball swings down to here, which gains momentum. Then it starts losing momentum as you come up here and eventually hits point B and stops. So at point L, you're going to have the greatest amount of energy or speed that goes into this point here. It increases to here, and then now it's going to start decreasing. We're going to look for an answer choice that says the same thing as what you just see there. Simply scroll down to the answers and take a look at 29. The ball's kinetic energy at point L is going to be greater because the velocity is greater at point L. A is the correct answer. Let's go ahead and see why some of the other ones are wrong. Let's start with the easy ones. D. The kinetic energy at point L is less because the velocity is less. No, the velocity is the most, and you saw that when something swings from here to here, this is the greatest amount of energy right in the middle. Then it's going to start going up and lose energy because it's like going up a hill. D is out for that reason. Let's go ahead and take a look at C. C says less because V is greater at that point. Well, if you have your pendulum here and the ball, and we start it up here and let this go this way, the energy is not going to be the least because this is where the most velocity is and the most power is. So that's out as well. Let's take a look at B. B says greater because V is less. Well, you know, V, the velocity is not less because the velocity is the greatest right here. Remember, going down a hill, your velocity will be the greatest, and then now you're going up a hill. A is the correct answer for 29. Test 68C, Science, Practice Problem number 30. Number 30 reads, based on student 3's description, when the ball is moving away from the wall and is slowing down, PE is accumulating. Where is the accumulating PE being stored? A couple things here. First of all, we need to go take a look at what's going on in student 3, number 1. Number 2, we need to go ahead and find a definition for PE. So let's go ahead and do that. Finger scan from the top of the passage to find out what PE means. When you come up here in the mini introductory paragraph, paragraph 6, we look for PE. We find PE right here, and PE says potential energy. Okay, let's go ahead and take a look at student 3, and when we find student 3 here, you see student 3 is dealing with figure 3, so we understand the setup. In figure 3, we have an overview, a wall, a table right next to the wall, a ball on a string, and the string is being stretched. So think of that string as being like a rubber band. Here's what they're going to do. They're going to go ahead and pull this ball this way, stretch the string, and then the energy is going to shoot it towards the wall. Here's what they're saying. Once the ball hits the wall and starts going this direction again, this string might be loose. For example, if this ball is here, this string is kind of loose, still attached to the wall. Where, in this scenario, is the potential energy, PE, being stored? Let's go ahead and take a look at some of the answer choices. And you see that it says F in the ball is the string the ball or the table where the potential energy is. Remember, here is the diagram. Here's the table and the wall. Here is that ball halfway out, and that string is kind of loose. The potential energy, is it in the ball? No. Ball is basically going to move. Is it the wall? The wall is stationary? No. Is it the table? The table is stationary? No. Its potential energy is in the string, because once this ball gets to a certain point and the string stretches out all the way to the end, guess what's going to happen? It's going to hit, it's going to stretch a little, and it's going to snap back the other direction. And the other direction making it go that way is solely due to the string, and therefore the potential energy for the return of this ball that direction is based on potential energy. Therefore, the correct answer, the energy, potential energy, will be stored in the string. 30 is J. Test 68C, Science, Practice Problem number 31. Number 31 states, throughout the ball's motion as described by student 1, does the amount of angular momentum of the ball, AM, remain constant? First thing that we need to do is we need to come in here and get a definition for AM, angular momentum. Then go back into student 1 and see if it remains constant. Let's go back up to the mini introductory paragraph that will give us all the definitions that we need. We can simply finger scan or get our eyes moving to find the correct area of AM, or angular momentum. AM, 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 we find it here. And the definition of the AM, the amount of angular momentum of the ball, AM, equals MVR, mass times the velocity, times r, and r says is the distance of the ball from the reference point. Let's go back into the question. And read it one more time. Throughout the motion described by student 1, does the angular momentum remain constant? Let's go back in and take a look at student 1's setup. You can find student 1's setup in figure 1. Here's student 1, and the setup is in figure 1. And let's go ahead and take a look at everything. First of all, does the mass of the ball change? No. Does the distance change? R. Here is R. Going from A to L to B. Does this change? No, because it's basically the length of the string. Does the velocity, when you go from here to here, the velocity will increase. It's like going down a hill. So V will be increasing. Now we're going to be going uphill to point B, so the velocity will be decreasing. Simply looking at the figure. Now let's go back into the answer choices and see what we have. Will the angular momentum AM remain constant? No, because the definition we saw for angular momentum is mass times velocity times r, the distance. We know that the distance is not changing, the mass is not changing, but the velocity will be changing. So the correct answer is going to be no. It does not stay constant, not constant. A can be crossed out because it says it does stay constant. B says yes, it does stay constant. And we know the answer is no. So we're left with answer choice C and D. Let's take a look and see. No, because although M and V remain constant, no, V does not remain constant. Remember, you have a pendulum with a ball on it. A is here, L is here, and B is here. Going from A to L, we said is going to increase the velocity because we're going downhill. Then from L to B, we're going uphill, which means it's going to lose velocity. So that is not remaining constant and therefore can be crossed out. D says no because although M and R remain constant, yes, the string length, distance, remains the same and the mass of the ball remains the same. True. V does not remain constant. D is the correct answer for 31. Test 68C, Science, Practice Problem number 32. Number 32 states, based on student 2's description of the motion of the ball as R decreases, how does the ball's KE change? A couple things here. First of all, we'll need to know some definitions here. KE is the kinetic energy, which is basically the movement, how much it's moving. And R, we can go ahead and finger scan for that up in the top in the mini introductory paragraph. We come up here to passage 6. We look for R. You find R right here. And R says it's the distance from the ball to the reference point. Now, the question asks us about scientist 2 or student 2. Right here, that will refer us to figure 2. And in figure 2, you can kind of get a feel for what's going on here. Here's what's happening. There's a table. It's a person's hand underneath the table. There is a ball with a string attached to it, and the person is taking the string and pulling the string down, therefore reducing the length or reducing the R. Here's the hole, here's the ball, and think of this as the string continuing through the hole. If I were to take my hand here and pull this string down, this distance would decrease and the ball would eventually move this way towards the hole and stop once it got to the hole. So as the string, or R, is getting shorter, we're picking up speed until we finally hit the hole right here. Just by looking at that from figure 2, we kind of get a feel for what's going on here. Let's go back down to the answer choices here. Take a look one more time. We'll read that question real quick. It says, based on student 2's description of the motion of the ball, as R decreases, what happens to the KE, the kinetic energy? Well, you know that as R decreases, what's going to happen is the velocity, the speed of the ball is going to increase, as we said, towards the hole. So when KE is going to change, KE is going to increase. The kinetic energy or movement will increase. So it's going to increase, and it's solely going to increase because the velocity increases. Once again, look at that up here. You see the ball, and when you decrease this line here, the ball is going to speed up to the hole, and the kinetic energy, the movement, is going to increase because the ball is stationary and speeding up till it finally hits the hole when the string is underneath the table and no longer on the top of the table. Correct answer for this problem is going to be answer choice F, increase as V increases. Let's look at some of the other answer choices and see why. Answer choice G says the KE will increase because the velocity is decreasing. Well, that's not true. You know, the velocity is increasing. We saw that before. And answer choice H and J are both wrong because it says the kinetic energy is going to decrease. Kinetic energy basically means the movement, and it's not going to decrease because if you look at the experiment, the ball is stationary, which means the kinetic energy would be 0 at the start because it's sitting there, it's not moving. The only way KE is going to increase is when it starts to move. So you see those answer choices are both wrong. That will lead us to the only answer choice that's going to work here based upon what we've seen. It's answer choice F, increase and increasing because the velocity increases. 32F. Test 68C, Science, Practice Problem number 33. Number 33 states, speed V is momentarily 0 at various times during the motions described by which of the students? Let's quickly do an overview of all three of the students. You see that student 1 here is figure 1. In figure 1 we have ceiling, a string with a ball on it, and the ball is stopped. They let go. It swings to the bottom here. And then from the bottom, it swings back up here and is stopped again before it would swing backwards. So you see that we're starting here at stationary, which the velocity would equal 0. We swing here, which the velocity would be the greatest. And then back up here, we would hit B, and then V would equal 0 again at B. So student 1 is going to be in there. Let's go ahead and take a look at student 2. Student 2 deals with the table. Here is student 2. And let's look at the figure. We basically have a ball attached to a string. When you pull the ball, it goes down and stops. There's no point, at any point in this experiment, does the ball momentarily stay at velocity of 0. It either starts at 0 and ends at 0, but nowhere in between would it stop and have a velocity of 0, just like you saw in student 1, where the velocity would be 0 at other places besides L. B, it's hitting the top and then stopping before it returns back down, and here is stationary. So this is the key point here, B. It's momentarily in the process of being velocity of 0. Let's go ahead and take a look at student 3. And student 3 is right here, and the setup is from figure 3 right here. And here's what's happening. We have a table pushed up to a wall. There is a string attached to the wall. There is a ball attached to the string. The string is being stretched, and then the potential energy is shooting the ball towards the wall. Once the ball hits the wall, the movement stops. It's going to be on the wall. It stops. So that means once the ball touches the wall, the velocity would be 0. After it hits the wall, it's going to shoot and rebound off the wall in this direction. But you see that V is going to be 0 when it's touching the wall. So student 3 would be included in the answer as being yes. Student 2, there is no area where the momentary velocity is going to equal 0. So the correct answer is going to be student 1 and student 3. You can find that answer in answer choice C for problem 33. Test 68C, Science, practice problem number 34. Number 34 reads, suppose that TME is conserved throughout the ball's motion. Based on the information given, if TME equals 100 ergs, KE and PE can simultaneously have which of the following values? First things first, go back in, finger scan, find definitions for TME and for KE and PE. Best place to find that would be to start at the top in the mini introductory paragraph. When you go back to the top, we can simply look for those terms. We go back up, it's passage 6. We go ahead and find KE here is kinetic energy, and then we go ahead and find PE is potential energy, and our TME is this, total mechanical energy. And it tells us the definition of this. This is awesome. TME equals KE plus PE. Well, there's the answer. Scroll back down to the answers here, and it says, I would write that formula down, TME equals KE plus PE. Now, it gives us that TE equals 100. So that means 100 is going to have to equal something plus something. These two together, when you add them, KE and PE, are going to have to equal 100 because that's the definition of the total mechanical energy. Let's look at F. KE 10 plus PE 10, does that equal 100? No, it equals 20, so that's out. G, KE 10 plus 90, does that equal 100? Yes, and that's the right answer. Let's look at the other answers just to make sure you understand how we derive this. TME equals KE plus PE. So we have KE 125 plus PE 125 plus 25. Does that equal 100? No, that equals 150, so that's out. And J says 1,000 plus 10. That does not equal 100. So the correct answer here is basically the definition of TME, total mechanical energy, is coming from kinetic energy plus potential energy. That will equal both of those. And they are given the value of 100. So we need an answer that's going to equal 100. 10 plus 90 is 100. G for 34. Test 68C, Science, practice problem number 35. Number 35 states, suppose that when R equals 5 centimeters, V equals 20 centimeters per second. Based on the statement by student 2, when R equals 15 centimeters, V would equal the value closest to which of the following? The first thing that we need to do is we need to first of all get going and take a look at student 2 before we start monkeying around with the rest of this. Student 2, you see the setup, and student 2 has the table with the hole in it and the string. So here is student 2, and we're in figure 2 right here. Basically, you have a ball with a string on it, and the person is pulling the string down, decreasing the string length. As you decrease the string length, the speed of the ball will increase. So R goes down, V goes up. See what I just did here? Relationships. As R goes down, V will go up. And that makes sense because you pull that string, the ball is going to speed to the hole, and the string length is going to get shorter. The string length is going to be R. So it's all about relationships. The other thing is this. If you look at what it says here, it says this in this. The student says the speed V increases between the two measurements because product VR remains constant, but V will continue to increase as R decreases. That's exactly what we saw there. We already knew that. We didn't even need to read it, but it's another way for you to find out that information here. So let's go back into the question and weed through the details. Now we see that we have, let's write some stuff down. It says we start with an R of 20, and remember that as R decreases, V will increase. So we'll just write this up here, R and V. Or another way to say that is as the string increases, what do you think happens to the velocity? Same thing. The relationships are exactly the same, only flip-flopped. Here we have R equals 5 and V equals 20. Now they give us a new R value, which is larger. So you know that the velocity, when you increase the R, the velocity will have to go down. The velocity is going to have to be below 20. So look at your answer choices, and right away you know that 19 and 15 can be crossed out right away, because here we have R equals 5 and R equals 15. This is 3 times each other. So that means that V is going to have to be reduced by 3. 20 reduced by 3 would be roughly right around 5 or 6. Look at your answer choices, and C and D definitely can be crossed out. Which one is closer? When you reduce 20 divided by 3 gives you 7, because 7 times 3 is 21, so right around 7. B is the correct answer. It's a relationship question. Remember, use your arrows, plus, minus, to designate relationships. Everything in science is basically about relationships. 35, B. Test 68C, Science, Practice Problem number 36. Number 36 says, consider a solution for which C equals 0.2 moles per liter and pi equals 10 atm. Based on figure 1, this solution most likely is an aqueous solution of what? Here's what we need to do. Go ahead, look at figure 1, then take a look at the xy axis. The x is the C value, and the y is the pi value. And then basically take your fingers and trace along the borders. So you come over here. Here is figure 1 right here. And in figure 1, let's scroll down a little bit so I can get it on the whole screen. You see in the x-axis here we have C moles per liter. And now remember, we're going to plug in the information that we're looking for. We're looking for 0.2, which is here. Here. So we're going to go up and we're going to find 10 pi, which is the y. So we're going to scroll over here and very carefully find where those two lines meet. And you see that it's this line right here. If you look at this line, it's NaCl. And if we continued straight up the line here, you would see that this is 310 Kelvin. NaCl 310. That's our answer. Let's go down and see which one of those. It's G. That easy, kids. 36 G. Test 68C, Science Practice Problem number 37. Number 37 states, compare to the mass of 1 molecule of glucose, the mass of 1 molecule of lactose is what? We need to go and take a look and see, first of all, where they start talking about molecules of glucose and lactose. We can find that information when we scroll back up in Table 2. Table 2, we see Table 2. We have the molecular mass or molar mass of glucose and lactose, and it looks like the mass is 342 versus 180 for glucose. It looks like our I value is 1 and 1 for both of them. I don't know what I means, so I go back up here and try and figure out what that is real quick. If you look in Table 1, it gives you the definition of symbol I, the number of particles or molecules or ions formed per molecule. Okay, great. So now we know that they're basically the same, 1 and 1. Which one has a larger molar mass? You see lactose has a larger molar mass than glucose. Come on down to your answer choices and read the question. Compared to the mass of 1 molecule of glucose, lactose is what? Lactose, you saw, was heavier. Lactose 342, glucose 180. Lactose is heavier or larger. So that means answer choice A and B can be crossed out because it says lactose is smaller. No, lactose is not, because lactose is 342 and glucose is 180. 180. Sometimes kids like to write that information next to each one so you have it handy. You might use it in another question as well. A is out because it says smaller. B is out because it says smaller. 342 versus 180. You know that A and B are definitely out. Let's take a look at both of these, C and D. It says lactose is larger because lactose has a smaller molar mass. No, 342 versus 180. That is not smaller. C is out. D says lactose is larger because lactose has a larger molar mass than glucose. 342 versus 180. D, not a bad idea to take the information and write it right next to what you're looking at. You might use this somewhere else in the passage. 37 is D. Test 68C, Science, Practice Problem number 38. Number 38 reads, how does the value of I for HCl differ from the value of I for MgCl2? And what is the meaning of the difference? The value of I for HCl is what? First things first, we need to go ahead and take a look at I values for HCl and I values for MgCl2. We can go ahead and scroll on up and see that in table 2. First of all, we find HCl, and you see that HCl, the molar mass for 2 I's is 36, and the molar mass for 3 I's is 95.2. Now, I'm scratching my head saying, well, what is an I? I says this. We can find I in table 1, and it gives you the definition of I. Always when they give you unknown terms, go back and look and see what they are. The number of particles formed per molecule or unit of a substance when it is dissolved in water. That's important. All right, it's dissolved in water. Number of particles. So we could do a simple math calculation here and say we have 95. So for 3 of these particles dissolved, if we divide 95 by 3, you're looking at roughly 31, 32, 64, 96. All right, so this is roughly like 32. All right, just write that off to the side. One of these equals roughly 32 grams per mole. All right. Over here, HCl, it looks like you've got 2 equals 36. Well, 36 divided by 2 is 18 grams per mole. So you see that HCl is less massive per I. And remember, I is 1 dissolved in water. Let's go back down to the answer choices and see where we're at. Come over here, read the question again. It's longer than 2 lines long. Make sure you play around with the data and then read it again. How does the value of I for HCl differ from the value of MgCl? Well, the value of I is less. We saw that from above. Remember, we said 1 I was 18 for HCl, and one of these was around 32, so it's less. So the value of I for HCl is less or lower. We can go ahead and cross out answer choices that are opposite of that say higher. And H says higher, so that's out. And J says higher, that's out. Remember, HCl we said was 18. Just write it in. We might need it for later. MgCl2 was right around 32. So that leaves us with F and G. F says lower which means that each HCl dissolved in water to form fewer particles than does MgCl2, or lower, which means that each HCl dissolves in H2O to form more. Well, think about it this way. If you've got more stuff to dissolve here, your I value is lower, so you're going to have fewer. You're not sure about that? Go back into Table 1 and look at I again if you're unsure. Read what it says about I from Table 1. The number of particles, molecules, or ions formed per molecule or formula unit of a substance when it's dissolved in water. Right there, that leads you to answer choice F, because when you have an HCl and 1 equals 18 versus MgCl2 equals 32, it's going to be lower and it's going to be fewer particles. F. Keep in mind, you're trying to get the right answer, but try and watch what I'm doing here, kids. Notice I'm constantly going up and down, back and forth, putting this in my own words, writing notes to myself. Notice how I wrote over here off to the side, 18. I wrote off to the side over here, 32. When I come over into some of the other questions, I might not need to go back into the passage because I've got that stuff written down. It's right there, handy. It's again, 18. 38 is going to be answer choice F. Test 68C, Science, Practice Problem number 39. Number 39 states, suppose 1 liter solution is produced by dissolving 2 moles of KCl in H2O. Based on the information provided, pi in ATMs of this solution at 290 degrees Kelvin can be calculated by using which of the following expressions? The first thing that we need to know is we need to know what the definition of pi is, and that's some information from the very beginning up here. They give you pi here as being pi equals 0.0821 times t times I times c. So when you come over here and look at the answers, you know that our answer here, it's not dependent upon 0.0821 because they all have that same thing. It's not dependent on the temperature because they give you the temperature of 290. It is dependent on these two columns over here. So if you go back up to the formula, you see which one is the second column. Second column here is our I value. So we know that we can derive the answer to this problem by simply finding out what the I value is for KCl. It's either 1, 1, 2, or 3. Let's go back and find out what I equals for KCl. That information is also up here in Table 2. Here is our I value. We simply scroll down until we find HCl. HCl is here. It looks like our I value is 2. When we come back over here, the answer will be 0.0821 times 290 times 2, and we're done. C for 39. Test 68C, Science, practice problem number 40. Number 40 states, suppose a new line showing how pi varies for aqueous MgCl2 solution at 340 degrees Kelvin were added to figure 1. How would this new line compare to the lines shown in figure 1 for aqueous NaCl solution and aqueous glucose solutions at 340 degrees Kelvin? First things first, kids, this is a very long question stem. Make sure that you go in here and put it in your own words. Circle information that's important. Here is what we're going to do. We're going to go ahead, take a look at figure 1, and first of all, look at NaCl and look at glucose. And it tells us to look at them around the 340 mark. Let's go ahead, jump on up to figure 1. And when we go back up to figure 1, here is figure 1 here. And we will notice a couple things here. First of all, we have C for the X and pi for the Y value. We notice that we see glucose here in a cluster right down here around 5 pi. And then we have the second thing here, NaCl, right around here at a different level. It looks like the cluster is higher. Now, we wanted to know about 340. So we come over here, we see glucose, and we see our values range from 280 to 340. That comes over here, 15. Then we find NaCl again. We quickly look, we see 340 to 280 up here, and roughly around 30. That's interesting. Glucose, the area they're asking us about is 15 pi, and NaCl is double that. So there's something going on here. Let's go in and take a look at table 2 and look at the difference between glucose and NaCl. First of all, you see NaCl here, and it has an I value of 2. Then you look at glucose. Glucose has a value for I of 1. Well, when you double 1, what do you get? You get 2. What is the value of I? What does I mean? You can go up to table 1 here, find out what I means, and I means the number of particles or molecules formed in this when you're dissolving it in water. The number of particles. Well, that would make sense. Glucose, you only have 1 particle. 1 particle at 280 to 340 is 15 pi. And over here, when you double that, and now you have 2 particles, an I value of 2, you get double 15, which is 30. So what we've derived here is this. Glucose, I equals 1. NaCl, I equals 2. Go back to the problem and see what we want to do now. Here is we want to go ahead and take MgCl2 and add that in to figure 1 and see what happens. Come over here. We quickly look over at table 2, find MgCl2, and you see now that our I value is 3. That means 3 particles. If 1 particle at 340, I value 1 is 15, and 2 particles at 280 to 340 is 30, then an I value of 3 would probably be up here at 3 times 15 or 45, be up here. So we know that when we add our line in here, we are going to start down here at 0, but we are going to go up like this. It's going to be behind the NaCl. It's probably going to be more like up in here, in this region here, going up and up and up until we hit right around 45 up here. So probably somewhere in this area here. What's that going to do to NaCl? That's what they're asking. When we add in MgCl2 here behind what's going to happen to this line? Let's go look at the answer choices. And you see that when we go down to 40, what will happen, answer choice F says the y-intercept of NaCl and glucose will remain the same. Yep. But the slope of the line is going to be greater. Well, that makes sense because now we've added in one more line and the slope will go up. The correct answer, therefore, is going to be answer choice F. Take a look at some of the other answer choices and see why they're wrong. It says J, the y-intercept would be smaller for NaCl and glucose. The y-intercept is not going to change. So anytime J is out because it says it's going to be smaller, H is out, it says it's going to be greater. The y-intercept won't change. Here it says the y-intercept would remain the same, but the slope of the line would be smaller. Well, the slope of the line, you saw where we put in MgCl2, that is not going to make the slope smaller. It's going to make it larger. So the only answer that will work for 40 is problem is letter 8. Excuse me. F. For 40, once again, F.